Thank you so much. First of all, I would like to thank the organizers for the possibility to present our research here today, or at least a little part of it, which is, as it was mentioned before, is about direct neuronal reprogramming. And let's first see dementia worldwide in numbers. Currently, there are about 50 million affected people with dementia, which is going to be predicted to double in the next 20 years. And if you think about it, this is causing an enormous burden on our healthcare system, but also it's causing a society and economical problem. And what is behind dementia? I'm sure many of you know that there are many neurodegenerative diseases which are causing dementia, like Alzheimer's, Parkinson, Huntington's disease, or frontotemporal dementia, which is now getting more and more attention due to Bruce Willis. But there are also many other neurodegenerative disorders which are a little bit less known. And in a population of 50 million people, about 1.5 million people will be directly affected with neurodegenerative disorders. And as the population ages, this number is going to dramatically increase in the future. But obviously, there are many other people affected by dementia, the caretakers and the family as well. So neurodegeneration is a major healthcare crisis in the developed, but also in the developing world as well. And why is that? It's because there are no disease-modifying treatments available. And many of these diseases are currently uncurable, deadly diseases. So obviously, there is more and more need to understand neurodegenerative diseases and human aging as well. But why is that we couldn't solve this problem? Why is that there is no cure for these diseases? This is mainly from the huge complexity of the brain. The average adult brain contains about 10 on the 9th neurons, and each neuron has about 7,000 synaptic connections with other neurons. So if you think about when you want to find out what does not work properly in this system, this is very much analogous to finding a needle in a haystack. So then the questions that arises that how do we really test a drug before using it in clinical trials, before using it in humans? And actually, can we predict success with today's model at all? Why is that so important? Drug development from scratch is about $1–2 billion, but also it's not just the price of it, it's the time, how much time it takes to develop novel drugs. It's around 10 plus years, 15 years. So it's extremely important to be able to model aging and age-related neurodegenerative disorders. Here you can see the model systems that we mainly use in our lab. And I'm going to talk about a little bit the pros and cons of them. So you can see that animal models, obviously they are smaller in size, the brain, a mouse compared to a human. That causes less complexity, and a mouse, for example, have a shorter lifespan, about 1 to 2 years. And also it's important to highlight that while animal models gave us, and also is giving us, an enormous information about these diseases, and it's extremely important to use them, but still, Alzheimer's, Parkinson, and Huntington's disease, and many of the neurodegenerative disorders are human-specific disorders, so they're not present in animals. Then we also use human postmortem brain tissue, which is obviously limited in availability. It also gives us just a snapshot of the disease, and many times when we get these postmortem material, the cells of interest are already degraded, they're not present anymore. And obviously, there's no disease modification possible in this system, and there's no follow-up observations. Cellular models, which I'm going to talk about a little bit in detail, are human origin, they're accessible, and stem cell-derived neurons are very much used worldwide, but they lose the aging signature of the donor. So let's talk about stem cells a little bit. A pluripotent stem cell is very special because obviously it's capable of self-renewal, but it's also capable to differentiate into any type of cell in your body. And for many, many time, we actually thought that pluripotent stem cells, once they're terminally differentiated, there is no way backwards. And I do think, and would like to outline, that we did think about this very recently that this is the way, that once a pluripotent stem cell is terminally differentiated into a neuron, a fibroblast, a blood cell, there is no way backwards. And only in 2004, the Yamanaka factors have been introduced, and then we found out that, yes, we are capable to reprogram these cells into pluripotent stem cells, which are called induced pluripotent stem cells, and this has been rewarded with a Nobel Prize in 2012. So this is only a decade ago. So now we know that if we have a patient, we can make neurons from embryonic stem cells, we can derive neurons, but we can also take somatic cells, fibroblasts, or blood cells, and then we can make induced pluripotent stem cells and differentiate them into neurons. But from embryonic stem cells and induced pluripotent stem cells, the epigenetic marks, so the aging signatures, are erased. So basically, these are 0-year-old neurons. So there is quite hard to capture aging and age-related diseases in these model systems. And then less than a decade ago, direct neuronal reprogramming has been introduced, which actually bypasses this stem cell phase and then directly converts fibroblasts into neurons by transdifferentiation. These are called patient-derived induced neurons, or INs. You basically you harvest a skin biopsy, you start to culture fibroblast cells from healthy donors or from patients with different neurodegenerative disorders, and then by using viral vectors, you directly reprogram them without any stem cell phase, without any progenitor phase, into a neuron. These transdifferentiated neurons contain the genetic, but most importantly, the aging signatures of your donor. So basically, this model gives us a possibility to study cells which are human origin, patient-derived, and disease-specific neurons. Also, they maintain the age. We can study many patients at the same time, which are also can be very representative for the disease. It's reproducible, robust, quick, because it only takes 4 weeks from the fibroblast to make these neurons, and relatively cheap compared to, for example, iPS technology. In 2017, during my postdoc years, together with the Palmer lab, we have developed an all-in-one lentiviral vector. So only one lentiviral vector was capable to directly reprogram adult human dermal fibroblasts into neurons. You can see the vector here containing transcription factors and REST inhibition, and you can see here the red mature markers are present in the neurons, and also green tau is expressed in these cells. But also, they do resemble on a transcriptomic and proteomic profile like a neuronal cell. And since 2017, we have used this methodology in several different diseases, in several different countries, from several different sources, clinical sources. Here you can see a representative image of 3 healthy controls and 3 Huntington's disease patients' fibroblasts converted into neurons with low and high magnification. You can see a clear neuronal phenotype, but you can also see that the cells express these red and green mature neuronal markers, MAP2 and tau. And then let's see this a little bit in numbers. So around from 50,000 fibroblasts, we can get 20,000 neurons. So this is an efficient methodology to actually make downstream molecular analysis of these cells. What can we do with these cells? We can look them with immunocytochemistry, you can stain them, we can perform Western blot, we can perform patch clamp electrophysiology, bulk or single cell RNA sequencing, mass spectrometry, and DNA methylation to provide information about their epigenetic age, and also high content automated screening microscopy. So induced neurons keep the aging signature of the donor. Here you can see the age of the fibroblasts when they were biopsy that was taken, and here you can see the DNA methylation age, which is a methodology that you can define by doing DNA methylation array. And you can see here that from the controls, the healthy donors, there's a very strong correlation between the age at the biopsy and the DNA methylation age. But in the Huntington disease neurons, you can see an accelerated aging. And this is true for post-mortem neuronal samples as well. So in Alzheimer's, Parkinson's, and Huntington's disease, for example, we don't know yet why, but we have an accelerated aging, and this is also kept in these neurons, in these induced neurons. The other cool technique that we use is high-content automated microscopy. So basically what you can do is you can put in a plate, and then this microscope takes thousands and thousands of images in a completely automated way, and it's connected to a server that can automatically, unbiasedly analyze millions of cells and can define cell number, neuronal purity, conversion efficiency, but also it can define information about the morphology of these neurons on a cell-by-cell basis on a fluorescence intensity. And also this is important because now this gives us a possibility to perform high-throughput drug screening using these induced neurons in 96-well plates. And now the outstanding questions are, can we identify drugs that reverse disease-related phenotypes using these induced neurons? But I think the even more important question is that, does such drugs work better in human than those identified in other model systems? To tackle these questions, from the generous support of the NRDIO, we are now developing a new drug screening platform to study age-related cognitive disorders using induced neurons. So basically, we're taking a bunch of fibroblasts, converting them into neurons, and performing drug screening, and try to use it as a preclinical model. Many of our ongoing projects in the lab are focusing on the development of this platform by using reverse translational studies, validation, drug repurposing, drug screening, precision medicine, and we're also trying to rejuvenate the neurons, for example. And for the last few minutes, I'm just going to highlight one of our ongoing studies, which I think is extremely interesting, that together with the Cambridge University, where there is a FAL-HD clinical trial running just now. There are 18 Huntington patients and 8 controls who are taking a drug, felodipine, which has already been used as a blood pressure medicine. And basically, they are taking this medicine for 3, 6, and 12 months, and blood and CSF is taken from the patients and the healthy donors, healthy controls, and then autophagy, lysosomal degradation pathway, and also mutant Huntington expression is measured in these clinical trial samples. In parallel with this trial, at the Saint-Malvais University, we received fibroblasts from the same donors, from the cohort, and we have converted them into induced neurons, and then also treated them with felodipine to look at the morphology of the cells and also to assess autophagy in these induced neurons. And what is happening now is we're correlating our results with the clinical trial and then see if we could have predicted what they see in the patients, but also more importantly, could we in any way predict if the drug is going to work or it's not going to work? This is the million-dollar question, basically. And my last slide, I would like to just show you some summary that, so basically from a patient, you can take a skin biopsy, and then you can start to directly reprogram the fibroblasts into neurons, use it for disease modeling, drug screening, but also possibly in the future, we could transplant them back to the patients. And then I'm going to leave you with some sci-fi, which is not so sci-fi anymore, that even more translatable possibly would be to use direct reprogramming in vivo. So basically, to take the neurons or the non-neuronal cells in the brain, which are residing there, inject reprogramming factors, and then basically convert them to the cell types which are lost in neurodegenerative diseases, and therefore be able to cure the patients in the future. And with this, I am reaching to my most important slide. I would like to acknowledge everybody in my lab at the Saint-Malvais University, also at Lund University. I would like to thank all of my collaborators. I would like to thank our funding and the NTA, and I would like to thank for your attention.